So, hello everyone. We are the group 6 and we are going to report thrombosis. So, what is thrombosis? Thrombosis is the process of a blood clot, also known as a thrombus, forming in a blood vessel. This clot can block or obstruct blood flow in the affected area, as well as cause serious complications if the clot moves to a crucial part of the circulatory system, such as the brain or the lungs. It is normal for the body to produce clotting factors like platelets and fibrin when a blood vessel is injured, to prevent an excessive loss of blood from the body. If this effect is overproductive, it can obstruct the flow of blood and form an embolus that moves around the bloodstream. Thrombosis can broadly classified as either venous thrombosis or arterial thrombosis, according to where the thrombus presents in the body. Venous thrombosis occurs in the veins and is categorized further according to where it occurs, including deep vein thrombosis, portal vein thrombosis, renal vein thrombosis, jugular vein thrombosis, Budd-Chiari syndrome, Paget-Scooter disease, cerebral venous sinus thrombosis. Arterial thrombosis, also known as atherothrombosis due to its association with atheroma rupture, occurs in the arteries. The blood stasis caused by arterial fibrillation may also cause this type of thrombosis. So, what are the causes of thrombosis? Virchow's triad. As it has come to be known today, the triad consists of stasis, vessel damage, and hypercoagulability, and is used to describe the etiology and assess the risk of thrombosis, especially of deep vein thrombosis or DVT. Endothelium injury. Endothelium injury is accompanied by loss of protective molecules and expression of adhesive molecules, procoagulant activities, and mitogenetic factors, leading to development of thrombosis, smooth muscle cells migration, and proliferation, and atherosclerosis.
As previously discussed, thrombosis is the formation of solid mass, or thrombus, in an uninterrupted cardiovascular system from constituents of flowing blood. So, how is thrombus developed? In the development and pathogenesis of thrombosis, Varco's triad will come into discussion. There are three components of the Varco's triad, which includes endothelial injury, alteration of the blood flow, and hypercoagulability. These three are interconnected with each other. Both endothelial injury and alteration of the blood flow causes hypercoagulability, which can cause thrombosis. And endothelial injury can cause alteration of the blood flow, which indirectly cause the formation of thrombus. And alteration of the blood flow can also cause endothelial injury, which can contribute in the formation of thrombus. In order to understand why endothelial injury results in thrombosis, first we have to know the normal function of an intact endothelium. Endothelium lines are blood vessels, and their function is to protect the flowing blood from the thrombogenic effects of subendothelial tissues. Endothelial cells also release substances that prevents thrombosis. These antithrombotic factors include heparin-like substances, thrombomodulin, inhibitors of platelet aggregation, and tissue plasminogen activator. How do they work? Heparin-like substances and thrombomodulin protect against the unchecked action of thrombin. Heparin-like substances accelerates antithrombin 3 to inactivate thrombin, and it also inhibits other clotting factors. Note that antithrombin 3 is a protein in the blood that blocks abnormal blood clots from forming. Thrombomodulin converts thrombin to an activator of protein C, which is an important anticoagulant, and it also has antithrombotic properties. Inhibitors of platelet aggregation include prostacyclin and nitric oxide. When platelets are activated after endothelial injury, they are inhibited from adhering to the surrounding uninjured endothelium by endothelial prostacyclin and nitric oxide, thereby limiting thrombosis. Endothelial cell also produce the enzyme adenosine diphosphatase that degrades adenosine diphosphate and further inhibit platelet aggregation. Lastly, tissue plasminogen activator activates tissue plasminogen, which helps in the fibrinolytic activity, which also prevents thrombosis. The endothelial cell not only releases antithrombotic substances but also some prothrombotic substances, which includes thromboplastin, von Willebrand's factor, and inhibitors of tissue plasminogen. Thromboplastin activates the extrinsic clotting pathway, where it aids in the conversion of prothrombin to thrombin, which is a powerful platelet aggregator and also plays an important role in thrombosis. Inhibitors of plasminogen activator decrease fibrinolysis and therefore favor persistence of the thrombi. Von Willebrand's factor mediates in the adherence of platelet to the damaged vessel wall. In summary, normal endothelium is thromboresistant, but when injured, it promotes thrombosis. Intact endothelial cells inhibit platelet adherence and blood clotting. However, injury or activation of endothelial cells result in procoagulant activity that increases local clot formation. Next, we will be discussing alteration of blood flow. In a normal blood flow of an unruptured or uninterrupted cardiovascular system, the white blood cells and the red blood cells occupy the central column, and they are the more rapidly moving axial stream. The smaller platelets are carried in the more slowly moving axial stream outside the central column. When there is turbulence or stasis, the platelets will come in contact in the endothelium, which will have an effect in thrombus formation. Turbulence means an equal flow of blood, whereas stasis means slowing of the blood. Another mechanism by which turbulence and stasis can result in thrombus formation is they prevent flesh-flowing blood from diluting, prevent the inflow of clotting factor, and promote endothelial cell adhesion. The last component of the varicose triad that we will be discussing today is the hypercoagulability. Hypercoagulability usually plays a less significant role in thrombosis, but it is still an important factor. Hypercoagulability can be caused by the increased levels of fibrinogen, prothrombin, and factors 7, 8, and 10. Increased number of stickiness of the platelets, decreased levels of inhibitors such as prothrombin 3 and protein C, and also fibrinolysis can also cause hypercoagulability.
So, example for endothelium injury, trauma such as lacerations, contusions, ruptures, frequent puncture of veins for intravascular injections, toxins such as streptococci and erysipelas pylori that injure the endocardium, viruses such as cholera virus which injure the mesothelium or spleen, parasites such as Strongylosis vulgaris can damages the mesenteric artery in horse, invading tumors that injures the endothelium. Next is alteration in blood flow. Abnormal blood flow, stasis, and turbulence, or the alterations in normal blood flow, which can cause endothelial injury, aneurysms, aortic and arterial dilations, cause local turbulence, a dilated arterium, in the presence of the atrial fibrillation in a site of significant stasis that may provoke thrombus development. Example, chronic venous congestion, aneurysm, varicose vein, congestive heart failure, shallow breathing due to pulmonary diseases. Next is hypercoagulability. Hypercoagulability can be defined as the tendency to have thrombosis as a result of certain inherited and or acquired molecule defects. Clinical manifestation or hypercoagulability can be devastating and even lethal. So the examples of hypercoagulability are increase in the number of thrombocytes from partosition and severe surgical operations, increase in adhesiveness of platelets after parturitions and operations, decrease in heparin is when anticoagulant decreases, the chances of thrombosis increases, increased amount of plasma fibrogen and prothrombin, which when the trauma occurs when the plasma fibrogen and prothrombin increases, the chances of thrombosis also increases. Increase of viscosity of blood, in which hema concentration is when the plasma volume decreases, the RBC also increases. Sludging of blood when trauma, red cells get agglutinated intravascular, lipemia, abnormally high concentration, and emulsified fat.
Lastly, we will discuss about the pathogenesis of thrombosis. When the endothelium is disrupted or denuded, the subendothelial extracellular matrix is exposed, which results to platelet adhesion, platelet aggregation, release of tissue factors, and depletion of antithrombotic factors, and of course, this will lead to thrombus formation. However, thrombosis can occur even in the absence of disruption in the endothelium. Whenever there is an imbalance between antithrombotic and prothrombotic activities, where prothrombotic activities are in the upper hand, thrombus can develop. Thrombosis can occur anywhere in the uninterrupted cardiovascular system. The pathogenesis of thrombosis can be summarized in a simple structure. First, trauma or injury in the endothelium. Second, the endothelium becomes rough or is activated. Third, platelets get attached, and this is also what we call the adhesive property. Fourth, more platelets are attached at the right angle of the flow. And lastly, a plug or thrombosis formed. When there is a mass of platelet formed, this is what we call the white thrombus or the pale thrombus. When a mass of erythrocytes is formed, we call it the red thrombus.
Today I will be discussing about the variety of thrombi. Thrombi varies in location, color, and in position. The first location is the blood vascular system. It consists of the cardiac thrombi. The cardiac thrombi is located in the heart, and some are attached in the valves or the valvular thrombi, or walls or mural thrombi. This is most common in pigs when their valves are invaded with Streptococcus pyogenes or Erysipelotrix rochefort. The second one is the arterial thrombi. The arterial thrombi is located in the arteries, and it is usually attached firmly in the damaged artery. This is most common in domestic animals, and it is less than common in their counterparts. The third thrombi is the venous thrombi. The venous thrombi is located in the veins. They are also called phlebothromboses. These are not usually common in animals, but it is very common in humans. It is not common in animals because when animals are ill, they still move around and stretch and move about, thus preventing the slow circulation in the damaged vein. Humans, in the other hand, will rest and go to bed when ill, we tend to lie down and try to recover. Uh, then this causes some slow circulation in the vein. This is sometimes seen in the vascular sinuses of cows and horses, also in the broad ligament of cows, and in the scrotal plexus of horses. It is often attached to underlying vessel wall. The next one is the capillary thrombi. The capillary thrombi is located in the capillaries, and it is mostly associated with inflammation. Because the etiological agents is easily injured by the vascular endothelium. Lymphatic thrombi, located in the blood vessel and sinuses of lymph nodes. Um, the lymphatic thrombi are composed of fibrin. They are useful because this thrombi prevents the spread of infectious agent in the lymphatic system. The next thrombi are located in the heart or the blood vessels. First, mural thrombi. They are attached to the wall of the heart or blood vessel. They occur generally within the cardiac chambers of or the aorta. Then, valvular thrombi, attached on the heart valves. And then, lateral thrombi, which are attached on the side of the vessel wall. Next is the occluding thrombi, which are attached to the entire circumference of the vessel. Then the saddle thrombi. They are found sitting in the bifurcation of blood vessels like a horse's saddle. Then canalized thrombi. Thrombi where new blood channels have been formed through the clot. The channels formed will partially restore the blood circulation and they are lined with the endothelium. The next variety is the infectious agent. Septic thrombi. The septic thrombi contains bacteria. Then parasitic thrombi. This contains parasites. Examples are Dirophilia immitis in dogs and Strongylus vulgaris larvae in horses. Then aseptic thrombi. They do not contain bacteria or parasites. Then the next variation of thrombi is color. Pale or white thrombus are almost entirely composed of platelets. It is seen on the heart and blood vessels, aorta, carotid, and femoral arteries. Then red thrombus are composed of platelets, fibrin, erythrocytes, and leukocytes. The shade red is caused by erythrocytes who are trapped in the fibrin meshwork. It is seen on veins and it is really important that we don't mistake it as postmortem clot. Then next is the mixed thrombus. This is the most common type and it is composed of white and red thrombi. The white portion, that means the blood flow is fast, while the red is slow. Then laminated thrombus. It is mixed thrombus with alternating layers of polar platelets mixed with some fibrin. It is separated with dark layers that contains more red cells, and laminated thrombus is the result of variations in the rapidity of blood flow. A white thrombus is formed when the blood flow is rapid, and the red is formed when it is slow. The next I will be discussing is the fate of thrombus. The first one is the propagation. It is the accumulation of more platelets and fibrin, or the vessel obstruction. Then emboli formation, it dislodges and travel to other sites. Then abscessation, abscesses appear on the thrombi when pyogenic bacteria are present. Then dissolution. Dissolution is removed by the fibrinolytic activity. Then organization and recalcination is the last one. This may induce inflammation and fibrosis, will reestablish vascular flow.
Effects of thrombosis. Negligible effects. No effects are noticed when thrombi occur in vessels which are not required. Experimentally, both jugular veins and both carotid arteries can be ligated in the dog with no adverse effects if the animals are not forced to move about. Beneficial effects. The primary hemostatic plug assists in the control of hemorrhage and is life-saving. Harmful effects. The harmful effects depend on which vessel is thrombosed and on collateral circulation in the area. Thrombosis in the end arteries result in infarction. This is commonly observed in coronary arteries in humans. Portions of the thrombi may break, float in the bloodstream as emboli, get lodged in the smaller arteries and cause infarction. Thrombi in veins cause local passive hyperemia. When lymphaticks are thrombosed, hypoedema of the area drained by the lymph vessel occurs. Thrombosis weakens the vessel wall, and this may cause its local dilatation known as aneurysm. This is observed in the anterior mesenteric artery of the horse following an invasion of strongyle larvae. Differentiation of postmortem clots from venous thrombi. A thrombus must not be confused with a postmortem clot. Postmortem clots have two types. First is the red or currant jelly clots, which is the components of the blood are evenly distributed. This occurs when there is rapid clotting of blood and its composition is normal. The second one is yellow or chicken fat clots, which is the components of the blood are not evenly distributed. The ventral portion of the clot is red and contains erythrocytes, while the dorsal portion is yellow and is composed of fibrin and plasma. These clots occur when erythrocytes do not remain in suspension and settle out. Example, in prolonged clotting time of the blood and increased erythrocyte sedimentation rate. Comparison of a thrombus and a postmortem clot. Thrombus. The characteristics of thrombus are the following: granularly dry and rough, white or pale in color, attached to the vessel wall, stratified in its structure, composed mainly of platelets. Vascular endothelium below the thrombus is damaged and rough. Forms in a flowing stream of blood. Forms in the living animal. May be partially organized. And lastly, caused by endothelial injury. Postmortem clot. These are the characteristics of postmortem clot. A rubbery gelatinous coagulum, intense red or yellow in color, not attached to the vessel wall, uniform in structure, composed mainly of fibrin. Vascular endothelium below the cloth is undamaged, smooth and glistening. Forms in a stagnant column of blood. Forms in the dead animal. No indication of organization. And lastly, initiated by thromboplastin.